Mechanism of enzyme action. Enzymes are complex nitrogenous organic compounds which are produced by living plants and animals. They are actually protein molecules of high molecular mass. These biological molecules are responsible for the thousands of chemical interconversions that sustain life. These include conversion of sucrose into glucose and fructose, conversion of glucose into ethyl alcohol and carbon dioxide, decomposition of urea into ammonia and carbon dioxide, etc. Enzymes speed up various biochemical reactions without being consumed in the reactions by following a proper mechanism. Let us discuss the mechanism in detail. The enzyme's catalyzed chemical reactions proceed in two steps. Step 1 involves binding of the enzyme to substrate to form an activated complex. The basic mechanism begins with the binding of the substance to the active site on the enzyme. The active site is the specific region of the enzyme which combines with the substrate. The active site has a unique geometric shape that is complementary to the geometric shape of a substrate molecule. The binding of substrate with enzyme is often compared with the lock and key mechanism. Step 2 involves the decomposition of the activated complex to form a product. The enzyme undergoes a slight change in its orientation to fit into the substrate perfectly. The binding of the substrate to the enzyme causes a change in the chemical bonds of the substrate and ultimately causes the reactions that lead to the formation of products. The products are released from the enzyme surface to regenerate the enzyme for another reaction cycle. Let us understand the mechanism of enzyme action more extensively by taking the example of hydrolysis of sucrose. The catalytic hydrolysis of the common disaccharide sucrose into the monosaccharides glucose and fructose is carried out in the presence of the enzymes sucrase. When sucrose binds to the active site of sucrase, the configuration of sucrase is changed. As a result, the oxygen bridge between the two monosaccharides gets exposed to water molecules in solvent. The water molecule breaks the oxygen bridge and the OH group of water molecule gets attached to one of the monosaccharides and the H gets attached to the oxygen of the second monosaccharide. Thus, the disaccharide is hydrolyzed into two separate sugars. On the completion of hydrolysis, the enzyme configuration is changed back to the original shape and the active site becomes available for other sucrose to bind. This action can be repeated many times until the enzymes become denatured or gets inhibited. The mechanism of enzyme catalysis is similar in principle to the other types of chemical catalysis. By providing an alternative reaction route, the enzyme reduces the energy required to reach the highest energy transition state of the reaction. The reduction of activation energy increases the number of reactant molecules with enough energy to reach the activation energy and form the product. As a result, products are formed faster and reactions reach their equilibrium state more rapidly. Most enzyme reaction rates are millions of times faster than those of comparable uncatalyzed reactions.